next test that we're going to talk about for Lab Unit 2, which is all about identifying gram-positive cocci organisms, is the coagulase test. Now, just by looking at the name of this test, you'll see coagulase, and you'll notice that ASE. That lets you know that we're dealing with an enzyme. So the purpose of this test is going to be to differentiate between organisms that have the enzyme coagulase and those that do not have the enzyme coagulase. So this test is strictly differential. Okay? Anything and everything can grow using this media. It's just going to grow differently based on whether or not it has the enzyme coagulase. So coagulase is an exoenzyme that many pathogens use for a variety of reasons. So we consider coagulase to be a virulence factor. So coagulase can cause clotting to happen. So you'll notice on the second bullet here that we use a media that includes rabbit plasma that has fibrinogen in it, which is a clotting protein. So if an organism has the enzyme coagulase, it will react with prothrombin in the plasma, and that is going to result in a complex formation called staphylothrombin. And this lets the enzyme convert fibrinogen to fibrin. Okay, and so that is going to be what causes the media to clot. So coagulation is like clotting. That's why this enzyme is called coagulase, because if the enzyme is present, you're going to see clotting. So coagulase, as I mentioned, is a virulence factor used by some pathogens. And the reason forming clots can be helpful is that some pathogens can actually multiply and replicate inside of that blood clot. And this allows them to replicate into large numbers without being detected by the immune system. Then once the pathogen has replicated and replicated, and there's tons of them, it can dissolve the blood clot using something like a hemolysin, which we'll learn about in our blood agar plate discussion. And then suddenly tons of pathogens are released into the body and can overwhelm the immune system. So it's kind of a sneaky tactic to replicate in secret. Another pathogen that we've talked a little bit about, Staphylococcus aureus, can actually coat its surface with fibrin, and that makes it hard for phagocytic cells to grab hold of it, ingest it, and digest it. So it's helping this particular pathogen evade our non-specific immune responses. So pretty sneaky. So let's take a look at what this media generally looks like and what will happen if you add a bacteria that has coagulase and what will happen if you add a bacteria that does not. There we go. Come on, computer. There we go. You can do it. All right. So when you look at this picture, look at the bottom tube. Do you see how it's a liquid when tilted? That's what the media typically looks like. Okay, when you grab the media to do this lab, it will be a liquid. So all you'll have to do is get some bacteria on your inoculating loop and swish it around in this media. You then place it in the incubator and you wait about 24 to 48 hours. And when you take these tubes out of the incubator, you're going to tilt them at an angle. If the media is still a liquid, that means nothing happened. There was no reaction. Okay, so that's a negative result, kind of on the bottom portion of the text on this slide. So it's a negative result because nothing happened, because the enzyme wasn't present. Okay, so think about what I just said and think about what is the observation, the result, and the interpretation. I intentionally left the interpretation blank because that's where a lot of students struggle, but I've actually already given you the answer in my words that I have spoken. So remember, your observation is what you physically see. We cannot see an enzyme. We cannot see a specific reaction happening, but we can see clotting or lack of clotting. So in this case, because the media is still a liquid, your observation is there's no clotting. Again, that's a negative result, and that tells us, hint your interpretation, that the organism does not have the enzyme coagulase. And it turns out Staphylococcus epidermidis is an organism that would give these observations and results when you inoculate this media in the lab. Now, if on the other hand, you inoculate Staphylococcus aureus into this media containing rabbit plasma, you will observe clotting. That's the top tube in this picture. So this would be a positive reaction because a change has happened, right? The media went from liquid to solid. So a reaction has occurred. That's a positive reaction. And that tells us that this organism does produce the enzyme coagulase. So the coagulase test and mannitol salt agar media allows us to differentiate between our two Staphylococci organisms. Okay, so Staphylococcus aureus does have coagulase, so it does form clotting, which gives us a positive result. And Staphylococcus epidermidis does not have coagulase, so it does not cause the media to clot. So we say it is a negative reaction. All right, y'all, that's it for the coagulase test. Let me know if you have any questions.